about take a space.
Presentation.
Yeah, we can.
Okay. Yes. So, hello, my name is Jonathan, and I'm here to give a presentation on my bachelor thesis, which is titled Modulation of Lipid Content in Beta Cells by Odd-Chain Fatty Acids. So, to start with, the World Health Organization released its first massive global report on diabetes in 2016, which stated that an estimated 422 million adults suffered from diabetes in 2014, and that diabetes caused 1.5 million deaths in 2012. In addition, the report declared that the global prevalence of diabetes in 2014 had doubled since 1980. Now, these remarks emphasize the importance of gaining further insight into the underlying causes of diabetes due to its huge burden on modern society, and that this disease shows no real indication of declining in the near future. So, diabetes mellitus encapsulates a group of metabolic diseases that lead to abnormal high levels of glucose in the blood. Now, within this group of metabolic diseases, type 2 diabetes stands out as the major cause of diabetes among patients. So the onset of type 2 diabetes has been suggested to associate with a decrease in insulin sensitivity, which leads to an unsatisfactory uptake of glucose from the blood. Now, insulin regulates the body's glucose uptake and is secreted by the beta cells, which is found in the pancreas. So it's possible for the beta cells to actually compensate for this decrease in insulin sensitivity by an increased insulin production. However, certain individuals are actually unable to compensate for this increased insulin demand, which leads to them developing type 2 diabetes. So these observations make it quite clear that the beta cell failure plays a pivotal role in the development of type 2 diabetes, but little is actually known about the underlying metabolic pathways that lead to this state of insufficient insulin response, in particular the lipid metabolism that occurs in the beta cell, where actually a lot of basic knowledge is relatively unknown. So, insight into metabolites that might play a key role in the development of type 2 diabetes can be obtained from previously conducted epidemiology studies, which has looked at the levels of various metabolites found in blood plasma. So certain studies have shown that a decrease of the odd-chain saturated fatty acids, abbreviated OCFAs, in the blood is associated with insulin resistance, while an increased incorporation of these OCFAs into plasma phospholipids has been associated with a lowered future risk of developing type 2 diabetes. And this kind of leads us into the aims of this study, which was to examine the lipid content in beta cells and how this is impacted by culture at elevated levels of these OCFAs. Now, to facilitate these studies, we also investigated the impact of sample amount on method performance by culturing cells on 6, 12, and 24 well plates, and also investigating the signal response obtained from UHPLC in non-LC compared to investigating the signal response obtained from non-LC in comparison to UHPLC. But before moving on to methods and results, I just want to briefly discuss some theory related to lipid structures. So, the hydrophobic property of lipids is in most cases derived from the fatty acids, which are a group of molecules with a hydrocarbon chain that varies in both length and degrees of unsaturation and is terminated by a carboxylic acid group. So on the slide here, we can see this general structure of the fatty acid and also some examples. So at the bottom of the slide, we can see the structure of palmitic acid, C16, margaric acid, C17, and stearic acid, C18. Now, these fatty acids can be used as building blocks to form more complex lipids, such as the phospholipids. So the phospholipids can incorporate one or two of these fatty acids into the phospholipid structure by connecting them to a phosphate group through a glycerol backbone structure, which is depicted here on the slide. Now, if we focus on the phosphate group, we can see that it, in turn, can be connected to different residue structures, which actually forms the various subclasses of phospholipids. So examples of these subclasses would be phosphatidylethanolamine, PE, phosphatidylcholine, PC, etc. Now, these subclasses can in turn undergo a hydrolysis process in which one of the fatty acids is removed from the structure. So, for example, if a PC would undergo this process, it would lose one of its fatty acids, and it would result in the formation of a lyso-PC, abbreviated LPC. So now we can look at the methods that have been used in this study, and here we can see a brief overview of the experimental procedure. So first, ENS1 832.13 cells were cultured on 6, 12, and 24 well plates, and then treated with either C16, C17, or C18 fatty acids at concentrations 0, 100, 200, or 400 micromolar, and in this case 0 micromolar is the control samples. After treatment, the cells were lysed and the lipids were extracted through a two-phase liquid-liquid extraction procedure, and then finally analyzed with a LC-MS system. In this case, we used a QTOF mass spectrometer fitted with an ESI ionization source, and the LC system was swapped between non-LC chip cube and UHPLC. So now we can look at some of the results related to the method performance. And on the slide here, we can see a chromatogram depicting the lipid PE361, and the peak that corresponds to a specific well plate size is marked. So what we can note here is that the signal intensity increases as we go from 24 well to 6 well plates. Now, this dependency is of course expected because the number of cells that can be cultured per well increases as we go from 24 well to 12 well to 6 well plates. Now, the far more interesting result here is that the samples taken from cells cultured on a 6 well plate didn't show any saturation effects on our LC-MS system. Now, this actually opens up the possibility to culture cells on a 6 well plate to increase the detectability of more rare lipids, which might otherwise which we would otherwise not be able to detect if we were to culture on a 24 well or 12 well plate. So moving on to comparing different LC-MS systems, on the slide here we see a chromatogram depicting the lipid LPC18O, and the peak that corresponds to a specific LC system is marked. So what we can see here is that the non-LC chip cube achieves a higher intensity at 1/4 of the injection volume used in the UHPLC system. However, due to time constraints, we couldn't really put an exact number how much more efficient the non-LC chip cube system is in comparison to UHPLC. Nonetheless, this chromatogram shows that the non-LC chip-Q system shows promise in further improving lipid detection, while at the same time consuming less of our sample in the process. So that kind of concludes results related to the method performance. Now we move on to results related to the lipid content in the beta cell. So on the slide here, we can see a chromatogram showing all the 28 different lipids which we were able to detect, and the majority of them were actually found in control and treatment groups, with the exception of LPC17O and LPE17O, which were only detected in cells treated with the C17 fatty acid. So to further investigate the impact of our treatments on the lipid content in the cell, we need to use statistical analysis. So on the slide here, we see the scores taken from a principal component analysis on the different fatty acid treatments and control. So from this principal component analysis, if we start to look at the scores taken from the second principal component, T2, it seems that the samples which were treated with C16 and C18 seem to be kind of differing from the control samples, which hints at these treatments actually having an impact on the lipid content in the cell. If we shift our focus towards the cells which were treated with C17 fatty acid, they don't really seem to be differing all that much from the control samples along the second principal component, T2. But if we shift our focus towards the first principal component, T1, the cells treated with C17 seem to be differing from all other conditions. Now, these clustering differences were further examined with conducting an ANOVA and a post-hoc test on these scores, and the results here are shown on the slide. So here we can see, if we start with the scores taken from the first principal component T1, we can see that all conditions are indeed differing from the cells treated with C17. If we shift our focus towards the other figure, which shows the scores taken from the second principal component, T2, we can see that all conditions are differing from each other, except between the cells treated with C17 and control. So the impact of our treatment can be further investigated by conducting a orthogonal projection to latent structures discriminant analysis, abbreviated OPLSDA, and plotting some shared and unique structure plots, or also called SUS plots. So on the slide here we can see such a SUS plot in which we are comparing each fatty acid treatment to the control. So along the horizontal axis we can see that comparing the C16 treatment group to control, and along the vertical axis we can see the C17 group versus control. So if we start along the horizontal axis, we can see that there are some lipids which seem to have a higher concentration in the cells treated with C16 compared to control, while other lipids seem to have a lower concentration in the treated cells. On further inspection, we can also see that the lipids which have a higher concentration, or seem to have a higher concentration in the cells treated with C16, are also known to incorporate one or two of the C16 fatty acids into their structure. And I marked LPC16O and PC32-1 as an example of this. If we now shift our focus towards the vertical axis, we can see that the majority of lipids seem to have a lower concentration in the cells treated with C17 compared to control, with the sole exception of PC35-1, which this lipid, PC35-1, might be derived from a combined incorporation of a C17 fatty acid and a C18 fatty acid. So this is just another SUS plot, but the only difference here is that we have C18 versus control along the horizontal axis. And the same thing here is shown, that certain lipids seem to have a higher concentration in the cells treated with C18 compared to control, and other lipids seem to have a lower concentration in the cells treated with C18. And once again, we can see that the lipids that seem to have a higher concentration in the cells treated with the fatty acid C18 are also known to incorporate the C18 fatty acid into their structure. So I marked LPC18O, P36-1, and PC36-1 as examples of this. We can also use ANOVA and post-hoc tests to look at how the fatty acid treatments are impacting the relative lipid levels. So on the slide here, we can see ANOVA showing the relative lipid concentrations between the treatment cell compared to control. Now, these relative lipid levels are only shown for the lipids that showed a significant difference in the post-hoc test between the concentration in the treatment group compared to the concentration in the control group. Those lipids that did not show this significant difference in concentration are just marked NS, not significant. Now, the first thing we can note here is that a majority of the lipids seem to have a lower concentration in the treatment groups compared to control, which might indicate that our fatty acid treatments have some sort of toxic effect on our ENS1 832.13 cells. But, of course, we can see that some cells indeed have a higher concentration in the treatment cells compared to control. So in the C16 group, I marked these lipids in green, and the C18 group, I marked these lipids in blue. For the cells treated with the fatty acid C17, it seems that it's really only PC35-1 which has a higher concentration in the cells treated with C17 compared to control. But of course, LPC17O and LPC17O also have a higher concentration in these cells. But since they were only detected in the cells treated with C17 and not detected in control or the other treatment groups, they are omitted from statistical analysis. So if we look at the lipids which have a higher concentration in the C16 and C18 treated cells, we can see that they are in line with the underlying theory that one or two of the same fatty acids are being incorporated into the same phospholipid structure, while again the PC35-1 lipid might be due to a combined incorporation of a C17 and a C18 fatty acid. Finally, we can also look at how the phospholipid levels are impacted by increased fatty acid concentration by using a linear model. So the regression model here shown on the slide indicates that a lot of lipids seem to have a negative correlation to increased fatty acid concentration or a non-significant correlation to increased fatty acid concentration, with the sole exception of 2 lipids marked here in green in the C16 group, which seem to show a positive correlation to increased fatty acid concentration. Now, the observation that a lot of lipids seem to have a negative correlation to increased fatty acid concentration sort of also supports this idea that the fatty acids have a toxic effect on our ENS1 832.13 cells. So, for conclusions, I've shown some data which supports the idea that fatty acids are being incorporated into more complex lipids. So we have seen examples of the C16, C17, and C18 fatty acids being incorporated into various lipid structures. A model system has also been created for studies of biological effects of OCFA-containing lipids. Now, what this means is that the data which has been presented here shows that the C17 fatty acid could be incorporated into the lipid LPC17O, LPE17O, and PC35-1. Now, what we could do then is that we could just add fatty acids to these cells, let these cells incorporate this fatty acid into these lipids, and then if we're interested to study if these lipids affect the beta cell function in any way, that's a way to go about it. We've also seen that it's possible to culture cells on a 6-well plate to improve detectability of more rare lipids, and that non-LC chip cube seems to be improving detection limits compared to UHPLC. So, future aspects. It's known that the branched chain amino acids can be metabolized into the odd chain saturated fatty acids in adipocyte cells. But if this occurs to a significant effect in other cells, such as the beta cells, remains to be explored. Another question could also arise then, if the branched chain amino acids are being metabolized into these odd chain saturated fatty acids, would it have the same effect on the lipid content as, for example, our C17 fatty acid? We could also extend our method to look at the incorporation of our fatty acid into other lipid structures, which we were unable to do within the time limit of this study. So, for example, we could look at if the C17 fatty acids are being incorporated into monoglycerides or triglycerides. And finally, we can look at the catabolic metabolism of the C17 fatty acids, how they are being broken, if they are being broken down in the cell, to what extent are they being broken down in the cell, and we can do this by looking at the acylcarnitine profile. Finally, I wish to thank Peter and Mahmoud for being great instructors. They have helped me a lot throughout this project. And here are the references, and thank you for listening.
Thank you. Very good. Thank you so much. So, questions?
Hopefully not too confusing. Sorry if it's really confusing.
No, it's absolutely not confusing.
It was very clear.
Yeah.
Great.
Yeah.
Huh? Do you have any questions?
Yeah, but I want to wait long.
You want to wait? Okay. Do you want to say something, Irene?
Yeah, I was
Huh?
thinking, one of the first things you showed us
Yeah.
about when you compare the 6-plate well and
You mean, let's see.
yeah, I think it was one of the first ones.
This one?
Yeah.
This one, yes.
Yeah, I was wondering what are the concentrations or what are you? Yeah.
The concentrations I'm comparing here is for 100. No, these are controls actually. So there are no added fatty acids here. This is just a control sample.
Mhm.
So they are control samples grown on 24-well plates, 12-well plates, and 6-well plates. So I don't know if familiar are with the plate sizes or these kind of things. Are you familiar at all?
We have
no
Okay,
idea.
I'm sorry.
I also
This
cannot
is important.
follow
Okay,
this.
this is important. So, for a 24-well plate,
Because it's very small.
they are very small wells. So means that you can't culture a lot of cells on such a well, in such a well. Meaning that you'll get less lipids when you extract, right?
Mhm.
But for a 6-well, these wells are huge. They are much larger than a 24-well plate. I think if we didn't do statistics on this, but the theoretical thing should be here that as we go from a 24-well to 12-well plate, there should be a 2-fold increase, kind of. And when we go from a 24-well plate to a 6-well plate, there should be a 6-fold increase. So that kind of tells us that there is more area in these wells to culture cells. We get more cells, and therefore we get more lipids. So that's basically what's being shown here. It's not concentration differences. It's this that in a 24 well you can culture more cells, or in a 6 well, sorry, you can culture more cells compared to a 24 well and compared to a 12 well. So
And then you extract more lipids,
Exactly.
and the amount of lipids that you get is enough for you to run the.
exactly, because that's why I also talked about this with the saturation effects in the LSMS system. The thing is, when you so we're running non-LC here. It's actually known that when you run non-LC, you can very easily run into these saturation effects, especially when you do it with an ESI ionization source in a QTOF system. But in this system, we didn't really see these saturation effects. So this opens up the possibility of just, if we're not seeing some rare lipids, which we know are just expressed really low in the cell, we could just culture more cells basically in a well, and then we just extract more of the lipids. But of course, there are other lipids which have a higher concentration in the cell, right? So we need to consider saturation effects there, right?
Because that is
But-
usually, I guess, very problematic, no?
Yeah, because there are some lipids are produced at a much larger rate, right, than others. So there's a lot more of certain lipids than other lipids in the cell. So that's the kind of problem because that's the kind of problem with culturing on a 6 well plate, right? Okay, we're increasing the concentration or the amount of lipids that we can extract from these rare lipids, but we're also increasing the concentration of the lipids that already are present at a very high concentration. So there are these issues with saturation effects, which in this case we were lucky enough that we didn't have them. I don't know if that answers the question or.
Saturation effect. Can you explain?
Yeah. So in a QTOF mass spectrometer, you have the detector, right? And then you have ions which are like ions hit the detector, that creates an electronic pulse, and then that is a reading, right? So when you have a saturation effect, you know that mass spectrometers have this dynamic range, right? It's a range where then you get a linear response if you increase like the ion count or the sample concentration. When you go outside this dynamic range, you get a nonlinear response. So what this means in this system is that if the ion count is too high, what you'll see is that you'll get a distortion of the amplitude of the peaks, and you'll get your mass accuracy, which you can the accuracy which you can determine masses in the mass spectrometer is totally out of whack. You can't really determine a mass with a reasonable accuracy anymore. So that's really what happens when you saturate the system. Does that explain it, or?
Yeah.
I think also with this, it's very difficult to know how many cells you can that is optimal, because very little is known about lipids in these cells. They don't store lipids and so on. So we don't really know. And then with the plates, if you culture cells in a 24 well plate, they might grow really nicely and confident, and then you put them in a 12 well plate, and all of a sudden they grow into colonies and behave differently. And if you go to a 15 centimeter plate, for instance, it's almost impossible to plate them. So you get all these kind of strange phenomena. So it's good
But
to
do
start
you
looking at.
weigh? I mean, if you have the wells, or you just add solvent to the well? Or how does it work? So do you know the mass of the cells that you? Do you know your sample weight? The sample
I
weight?
don't think we know that actually, really.
Just the amount of lipids extracted, maybe.
But they wouldn't. Would you know that?
I mean, I
Also
know that.
listen.
The general procedure here is that you culture your cells to a certain amount of confluency. Confluency is just that. So what you have is you have your well plate, right? And then you have the bottom of the well plate. The cells will attach to the bottom of the well plate and they will grow there. So what you basically do is that you let them grow until a certain amount of area of that area is covered with cells. So 100% confluency means that the whole area of that, the bottom of the well is covered. So I guess you could do some theoretical calculations there, but I don't know if we know the exact mass. You know?
And we will measure protein in the cells also. After extraction, to see how much protein as a proxy of cell numbers. But we did not do that now in the first. But I mean, it's a proxy. It's
Yeah.
I mean,
Yeah.
and there's also possibilities to count DNA, for instance. One set of DNA per cell, but that is sometimes not correct for clonal cell lines, which might have duplicates of chromosomes and stuff like that. And also, it doesn't tell you anything about the size of the cell, which might vary between conditions.
Yes, yeah, right.
So there's pros and cons for everything.
Mm.
But some things needs to be
Yeah.
done. I mean, eventually.
Yeah, no, in this case, I don't actually know. So, sorry.
Yeah. But you get the previous signal. That's the lipids, no?
Yeah, that's
the lipids that
that's
you
just an
start.
effect of that there's more
So you-
lipids.
Yeah.
So
You start with the same amount of solvent, so you just get a more concentrated.
yeah, so I mean, the extraction procedure is this, that you, regardless of if it is a 12 well, 24 well, or 6 well plate, the amount of organic and aqueous phase is the same. It
Yeah.
doesn't change. So basically, if there's more lipids in the lysed sample, more lipids are going to be extracted into the same volume of organic phase.
Yeah.
So then you'll get a more concentrated sample of your
That
lipid.
was the concentration that I was.
Yeah, you were wondering.
thinking.
Okay,
Yeah.
no, yeah, but
But, yeah, it's just more.
yeah, it's just more. That's basically it.
It's a relative, yeah.
Mhm.
And you mentioned that you get lower detection limits with the nano LC than with the UHPLC. Was that unexpected or?
No, I mean, when you compare UHPLC to non-LC, I mean, when you have very dilute samples, in non-LC you are lowering you're just you're making the column smaller, right? You're just going down to lower inner diameters of the column. So when you inject your sample, you get it more concentrated, basically, into the mobile phase. So, yes, it's kind of expected that you would get lower detection limits, absolutely. And we were actually seeing some results, I don't think it's with here, but some of the lipids which were actually not able to detect in UHPLC systems, for example, more rare lipids such as LPC15 and LPC17, just in control samples, not in cells treated with C17, we were actually able to see small peaks in the UHPLC system when we couldn't see anything in the non-LC system, when we couldn't see anything in the UHPLC system. So we would expect lower detection limits.
I also think there's a technical challenge also with running these chips with the pre-column, the concentrated column,
Yeah.
and to trap the lipids on that one. So there's many
Yeah.
additional challenges because this system is built for proteomics. So
The nano LC or
the nano LC
yeah.
chip. Yeah.
Okay.
So,
The chip, yeah.
yeah.
Can I ask? But what was the limit of detection, and limit of detection of what? I mean, is it
So
in the
this is
it
more-
the same solution you injected in both systems,
Yeah.
or is it the same sample? Or how did you
Um,
evaluate limit of detection?
the
I'm
thing
not
is
here
that-
to have to stop answering.
Sorry.
It's an examination.
Yeah, I know.
Yeah, no, no.
Last time you said that it was okay for me to speak
This
because I
is-
didn't say anything, but now I talk too much.
Oh, okay, it's okay.
But okay, so with the evaluation of limit of detection, as I stated, we didn't really do enough of evaluation to really determine the limit of detection, really. But I mean, I think we went. How to explain? So what we did is basically we had the same samples, right? The same control samples being extracted. And then we just injected some in the UHPLC, looked if we could see these more rare lipids. We couldn't, but then we injected them in the non-LC and we could see some of these rare lipids having this peak. I think it's 3 times the baseline or something, 3 times the noise, something like that, in chromatogram. So it wasn't really done in a rigorous way, so to speak. It was just that,
But you use the same sample.
yeah, we used the same samples.
But here you can see them in both. This is
Yeah, this is LPC18O. Yeah. But unfortunately, what I wanted to show here was actually, for example, LPC17O in a control sample. There, for example, you would have seen nothing, but in non-LC you would have seen a little peak.
But here you have the same concentration, what you inject.
Yes, I mean the same injection volume, concentration of lipid is taken from the same well plate size.
Yeah.
So it's controls which are taken from the same well plate size being injected into your HPLC and non-LC
yeah.
chip-cube system. So the concentration of the lipids should be the same.
Yeah, and this is repeatable. I lack a bit the analytical
Yeah,
quality
yeah,
things here.
absolutely.
Then maybe I can also.
Yeah,
we,
you
we
can
were
say
short
these
on
things,
we
it's
were
okay.
short on time because I mean this was installed. Yeah, first you had to prepare cells and then started running and some problems and I mean
Yeah,
stuff.
yeah,
So
yeah.
there were plans for more statistical evaluations and
Yeah,
to
okay,
generate the
okay,
data that
yeah.
was required.
That maybe will come later then.
Yeah, definitely.
You mentioned that it was important for you that you could use small injection volumes. What are the volumes of sample you're usually dealing with?
Oh, that's about, let's see now, about 400 microliters, something like that. So, but it depends also because, no wait, no it is 400 microliters. So basically we want to have a lower injection volume because we feel like it's a kind of limited to how much we're extracting from the liquid extraction protocol, but also that we can concentrate our samples more with just consuming less of our samples. Maybe it's not so important in this case, but it's just another it's just another way of observing that okay, we can use less of our sample, but we can still get a higher signal intensity.
So interesting, it differs so much since it's the same detector. Yeah. You didn't have any more questions?
No.
No? No? Okay. Maybe we should move over to more the chemometrics part to discuss those.
Can I have one question before we move over there?
Yeah?
It's probably in the report, or I missed it. Which chemistry of the column for the separation?
Oh, sorry? The chemistry, the stationary phase?
Yes.
Yeah, C18.
C18.
Yeah, for both columns.
Alright, cool beans. But I'm not a lipid separation guy whatsoever, but I'm curious, I've heard some people use silver-based columns for lipids.
This I've heard too, but I'm nobody to really ask about that because I don't know. But I've heard about it, yeah, too, this kind of separation.
No, it's just something I heard.
Yeah,
I was just
okay.
curious what
Yeah.
makes it
Isn't
better.
that in normal phase? Or?
Could be.
I think so. But
Like I said, I'm not a lipid separation kind of guy.
But
I
I
was
think
just
it's
curious.
old school.
Alright.
But it's nice to hear. You have silver ions in the...
This chip-based stuff is quite new, so they have C18.
Alright.
Okay, otherwise you can order special columns, but it's extremely expensive.
Alright.
So yeah.
That makes sense. Alright.
You didn't have any more question on this?
Not on this point.
No.
I'll save my stuff for
Okay.
second half.
Okay, so should we move over to more the chemometrics part? I'm wondering about all these plots, these PCA plots and these
LSDA plots.
SD plots.
Yeah.
For me, I find it very difficult. But one thing I find difficult is I don't get the idea of what the actual concentrations are. I mean, what are the absolute values and how you look at significant or non-significant differences. And but I don't have a feel for the real numbers.
Yeah.
And is it sort of, so I don't know if you know with this data if things are within a linear range, and if you look at very tiny little differences in concentration, or if they are really, because just because it's significant, maybe the absolute difference is so tiny. So if you have a plot and you see some clustering here and you see something there, if you would redo the whole experiment, do you think it would look the same kind of clustering here and something there?
Yeah,
the way it looks?
Exactly. So we did some repetitive measurements here, and we were indeed seeing like similar clusterings, but absolute values we didn't really look into that much.
No.
in this experiment. No.
So you don't know either what the concentrations are roughly and what differences we're talking about?
I mean, with these systems, it's quite minimal, I have to say, but exact concentrations, I don't actually know.
No.
Fortunately,
Yeah. Mm? That's
because
okay.
we could have done it, because basically you could have maybe done some kind of calibration where you looked at like, okay, how much, because what we did is basically we extracted peak areas, right?
Mm.
And that's basically the raw data.
Mm.
And then maybe we could have done some sort of calibration where we could see like, okay, how much is this in concentration
Yeah.
or something. But again, we were having some time limits, time
Yeah.
constraints. So
Because I could imagine that, for instance, if you have looked at different well size,
mm,
that's different sample amounts,
mm,
and I imagine you have a certain extraction solvent, and you will not do a complete extraction when you do like that.
Mm.
You will have a partitioning, and perhaps, or it should maybe depend on the sample size. I mean, how things are partitioning, you will reach distribution constant.
mm,
And so maybe it will affect whether you see or you don't see lipids, and it might affect your final results depending on if things go to completion or not, and
Yeah,
so on. I
exactly.
mean,
but we did.
So-
And
and
the
what
completion or not is like we did like vortexing and these kind of things also after. I don't know.
that improves mass transfer,
Yeah, that was mass transfer.
but you still have the thermodynamics
Yeah,
and
exactly.
equilibrium.
Yeah.
What I'm saying, so I'm just thinking that, but now this was a very short time you had. I
Yeah.
mean, so I know that. I'm really asking like it was much longer, but if you continue, I hope you will continue working on this later on. And I mean, because if you have cells here and you want to draw, your conclusion you draw down the road, you would like to discuss what happens in the cells.
Yeah.
Yeah. So then it's important that all the other things in between, the method,
Mm.
is doing what you expect it to do.
Yeah, exactly.
Yeah. So that's.
But I agree. I mean the analytical part here is a little bit lacking. Yes, I would agree. I mean, if I were to continue this, it would be more kind of thought out, maybe.
Yeah.
Absolutely.
Okay. Yeah. Okay. Did you have some question on the chemometrics?
Yes, of course. I mean, it's like if you use, a little bit more difficult cool methods, then of course you get a little bit more difficult questions as well.
Yeah.
Let's just go. I mean, the PSA, that's all nice and good.
This is okay, right?
Yeah, I mean, it's unsupervised method, so I mean, it
It's
is what it is.
the thing is that it's a bit hard to actually see here these kind of clustering distances which I am talking about. That's why I thought that this might kind of make it more clear.
No, I think this part for me is just really nice and tidy.
Okay.
But then we move to the next slide, and then we
Here.
start heading into the world of PLS.
Yes.
And this is where people usually get a little bit nervous. One is it's a little bit more difficult to comprehend. The second one, it's a very flexible method.
Mm.
So you're always, or at least whenever I'm doing PLS, I get a little bit nervous. Am I drawing the right conclusions or not from my data?
Mm.
So my super general question is, which validation methodologies or measurements have you done here to ensure that what you're seeing here is actually correct and not because of some fluke or random variation?
So I did not show these plots here, but we did do this kind of, when you have these dummy variables, right? When you try to separate very different classes. So what we were doing is, before we did this, we did a validation procedure where we started separating like the C16, the C18. Like for example here we're looking at a two-class comparison, C16 versus control. Okay, so what we do in PLS then is that we try to separate the scores for these two classes. So what we did before this is what we validated this by looking at, we drew basically a score plot where we looked at, are these two observations or are these two samples differing according to their group identity. So was C16 clumping in for itself, was a control forming clusters by itself along the what is it called? Because you have the orthogonal principal component, then you have the explanatory like principal component, the first one. So what we were looking at is basically, was the principal component that we were drawing here really differing these classes from each other? And that we did, and indeed it was showing that they were showing differences, like the first principal component was showing that they were clustering. Am I making sense here?
Yeah, I'm getting it.
Yeah.
I'm getting it. But just extending, I mean, I like this. It's a good way of approaching and thinking. But imagine that you have. Oh, by the way, one quick one. Did you use any scaling or what kind of pre-processing before finding away with the PLS?
The so everything was log transformed, the raw data, log
Yeah.
transformed, and then it's centered and scaled also.
All right.
Yeah, it should be, yes.
What's the motivation for the logarithmic
Well,
transformation?
usually biological systems usually have a skewed kind of, they don't really, because we want normal distribution, right? In biological systems, they usually have a skewed distribution. So we
Yeah.
log transform it to get a normal distribution. So
Yeah.
we can use it here.
Yeah, neat. I like it. But imagine, so you, just one last little case scenario. So imagine you have your two groups, your C16 versus your control, and then you have your some small peaks, which has some larger variation compared to some big peaks or whatever. Then imagine, for some weird reason, which I couldn't never be able to explain, there's this one peak or 2, 3 peaks, which is what separates the two groups, but it's just some major fluctuation or whatever. Or there's maybe, could be some sample that has some more influence than another one.
Yeah.
Could you think of some other neat statistical test? I know it's a difficult question, but it's a little bit important when you use PLS.
Yeah, I know.
You want to be like really certain.
Any other statistical test?
I think I know which one you talked about.
I'm a real big fan of these, so.
Is it the one starting with an O?
Oh?
O? No?
No,
No?
not sure,
Okay.
not sure.
I don't know if I can answer that question actually.
Well, there's so many tests and there's so many different philosophies, so I'm not gonna sit and say that one is better than another one, but I'm a big fan of resampling or subsampling. So because you have like a lot of repeated measurements, you have a lot of samples. I saw in the PCA you actually have quite a decent amount of samples, much better than most people in this field seem to have, so I'm impressed. But then you can actually remove a few samples, fit a model, see if it
Yeah.
predicts the other
Yeah,
ones, and then you can do that
exactly.
in different iterations. Or even better, you can have like a permutation test. So you say you have 16 in control, so you know the identity of your samples, right? What if you just switch the identities? So some samples have the wrong identity. Then your model should be wrong as well.
So you're talking about the predictability of our
Yeah,
model,
that
basically,
usually
right?
helps
Yeah,
convincing people, like me, if I were really skeptical. But it's a difficult question, but when you use a little bit more complex methods, it's
Yeah, no.
valid question. Nah, that was it.
Mm-hmm. Okay.
We did sevenfold cross-validation.
Yes, I like that.
It's automatically done.
Sounds good.
Okay.
We did it, that's how we calculated Q squared.
Yeah, exactly.
But,
Because I remember us looking
Oh,
at
I mean
the
outlier
predictability
test.
and the fit.
It's very
of our-
easy. Outlier.
Yeah.
Maybe not in this equation.
Well, I mean, yeah, we didn't really see any obvious outliers, so we
No.
didn't check for it.
No?
No.
Okay.
But yeah, it was definitely in the back of the mind.
That's the one that starts with an O. That's the outlier.
No, we didn't see any obvious outliers, so we didn't test for it.
Okay.
But actually, it's really difficult when you do PLS
Yeah,
to
yeah.
what is an outlier.
Yeah.
You end up in these gray cells and
Okay.
you're like scratching your head and spending weeks and weeks trying to figure out
Mm-hmm.
what you should do with it.
Mm-hmm. Yeah. Are there any other questions? No? Then we thank you so much for the
Thank you.
lovely
Thank you.
presentation. Thank you. So thank you all for coming. Mm?